Most of us think of our genes as fixed. That is, each of us is endowed with a genetic code that we inherited from our parents at birth. It's a lottery that we win or lose, with little that we can do about it. If it's in our genes, we imagine, we can't change it. That's why the idea that certain complex human traits are genetic can be dangerous. It makes you believe that if you're born with bad genes, you don't stand a chance. But this view of genetics is simply not true, as I alluded to in the last lecture. Yes, each of us has certain genetic predispositions, and genes are the basis for the constellation of traits that make us who we are. But the detailed study of heritability in genetics shows quite clearly that the environment and our experiences impact not only how we use our genes, but even the way that our genes are expressed. And there are many ways in which the DNA in our cells can change. In its extreme form, the idea that our genotype is fixed is the basis for genetic determinism, the notion that genes determine not just our characteristics, but our behavior too, leaving no room for free will. The presence of bad genes or the absence of good genes cannot be overcome with training, education, or otherwise, suggest the determinists. Take the 1997 science fiction film Gattaca, starring Ethan Hawke, as an example. The story takes place sometime in the future, and Ethan Hawke's character is a man whose professional prospects are limited by his genetic makeup. To overcome this limitation, he fakes his identity with the help of a genetically superior friend who provides blood samples, urine samples, and other biomarkers. I won't spoil the ending for you, but I will say that the movie portrays many of the complex psychological and societal issues that arise from linking genetics to behavior. And the implications are dark. Unfortunately, genetic determinism is not limited to the movies. In the late 19th and early 20th century, notions of biological determinism mixed with socio-political ideas of the time and contributed to the rise of the eugenics movement. The word eugenics means true genes, and its proponents sought to improve the gene pool by selectively breeding humans. The movement's intellectual roots trace to Sir Francis Galton, a relative of Charles Darwin as it happens, a man who made many important contributions to math and science. He theorized, quite benevolently, that an understanding of natural selection could eliminate unwanted traits or enhance desirable ones in us, just as it had in a variety of domesticated plants and animals, elevating our species and reducing our suffering. But the implementation of eugenics, most notoriously by the Nazis, was coercive and restrictive, arbitrarily targeting social groups based on prejudice. Atrocities such as forced sterilizations, euthanasia, institutionalization of the physically or mentally disabled, and ultimately genocide were justified in the name of improving the human gene pool. Lesser forms of racism are still very prevalent today and no less repugnant. They preserve the view that certain characteristics are inborn to racial or cultural groups. In a way, these practices persist because two psychological concepts collide in our minds: our tendency to categorize and generalize individuals into groups, and our intuitive notions that heritability lend a legacy to those group memberships. Take, for example, the notions of bloodlines and their importance in royal lineages in medieval times. Having royal blood made one special, superior to the lower classes. Or consider the one-drop rule in the southern US, which came to mean that a person was considered African American if they had even one drop of African blood in their ancestry, and back then, that was not a good thing. But the folly of these approaches is obvious when we begin to understand modern genetics. We now know that the idea that genes don't change is simply incorrect. Do our genes change across our lifespans? The answer is a resounding yes. And there are at least three major ways by which this happens: through chemical modifications called epigenetics, through sequence modifications called mutations, and through the insertion of new DNA as a result of viral infections. Let's consider each of these in turn. Recall that genes consist of a twisting double helix of DNA sequences. The DNA is not floating freely, but is tightly wound around spools of protein called histones at regular intervals. The combination of histones and DNA creates beads on a string, if you will, with the beads being nucleosomes. The strings of beads are then packaged and tied by other proteins and then wrapped and packaged again to create the wobbly, slim X shapes of the chromosomes that you might have seen in photographs. Yes, that's an oversimplification of chromosomal structure, of course, but I wanted to introduce you to histones and the proteins that bind to and interact with DNA, and the notion that DNA is packed and stored, like the strings of Christmas lights you might have in your attic. These ideas are important because they serve as the basis for the chemical modifications found in epigenetics. The dense packing of DNA within chromosomes saves a tremendous amount of space. It's been estimated that the full length of the DNA molecule in one chromosome, if unwound, would stretch about 5 centimeters or about 2 inches. That's 2,000 times the diameter of an average cell in our body, which is around 25 micrometers. Oh, and remember, each cell has 46 chromosomes, not just one. So that's a lot of DNA to pack into each cell. 3.2 billion base pairs, to be precise. But the clever packing serves another purpose, limiting the access of transcription machinery, the tools that turn genes into proteins. If the cellular transcription machinery cannot bind to the DNA, then the genes cannot be expressed. The proteins can't be manufactured. This is not a good or a bad thing, but a way of controlling whether or not a particular gene is expressed at a particular time. Gene expression starts with enzymes in the cell called histone acetyltransferases, affectionately referred to as HATs. HATs are responsible for linking an acetyl group, which is a small chemical addition, to the histone protein, which is the spool around which DNA is wound. Researchers first determined how HATs tack on the acetyl group, a process called acetylation, by observing their behavior in yeast. They figured out that this process leads to the activation of the genes in close proximity to that histone. How does it do this? The answer lies in part in alterations of the structure of DNA within the nucleosome, the beads along the string. What histone acetylation does is relax or loosen the DNA structure around the nucleosomes, a combination of histones and DNA, through a process called decondensation. Decondensation facilitates the transcription of genes by enzymes called RNA polymerases, which, as we saw in the last lecture, are responsible for the DNA transcription process. So hang your hat on a histone and your genes will come to life. Of course, Mother Nature doesn't usually turn on processes that she can't turn off. So there are natural foils to the hats called histone deacetylases, or HDACs. As the name suggests, HDACs remove the acetyl groups and in doing so promote the return of the gene into a silent state. So although the code from which the protein blueprints are read is relatively stable, if there are no hats lying around, the DNA structure can't be loosened enough to read off the instructions. This is one epigenetic way in which the internal environment of the cell can affect the expression of genes. A second form of epigenetic change is called DNA methylation, the process of linking molecules called methyl groups to the DNA nucleotides themselves. Note that this is subtly different from histone acetylation, which is the modification of the histone protein, not the DNA itself. And yes, there are enzymes to help us with the task, and they're called DNA methyltransferases, or DNMTs. What's the impact of DNMTs on gene expression? In general, they seem to suppress gene expression by physically impeding the binding of enzymes to the gene, for example, or via the actions of proteins that recruit other proteins, including HDACs, which we know act to silence gene expression. We are quickly discovering how important epigenetic changes are to human health and disease. Epigenetic modifications of DNA and the resulting alterations in gene expression are now recognized as being involved in several forms of cancer, for example. Many tumor suppressor genes, genes whose activity is lost when cells become cancerous, are silenced by DNA methylation. So to treat cancer, some labs are attempting to reverse methylation by inhibiting the DNMTs. Similarly, inhibition of histone deacetylases, a process that ultimately maintains histone acetylation, also promotes gene expression and has been shown to be an effective strategy for the treatment of certain forms of cancer. Epigenetic changes are reversible modifications of DNA that don't change the underlying genetic sequence. Changes in the genetic sequence itself do occur, however. The cells in our bodies are constantly replicating and renewing our tissues, as in repairing damaged skin after a scrape or a sunburn, or replacing the taste receptors on our tongue after a mild burn from a hot sip of tea. Now, each time a cell replicates, it must make a copy of its DNA. Cells aren't perfect. Neither are we, of course. So occasionally during the replication process, errors occur, permanently changing the genetic code in the new cells and all future cells born from it. These changes to the genetic sequence are called mutations, and their occurrence can have a range of effects on how the gene is read and how the gene product functions. And of course, as we age, these changes accumulate since we've had more opportunities to introduce mutations during cell replication. How do mutations occur? Some mutations occur naturally as a result of the imperfect replication process during cell division. As I mentioned earlier, there are 3.2 billion base pairs to copy in each cell, so even a very low error rate will result in mistakes here and there. In other cases, substances found in our environment, such as excessive UV radiation or carcinogens like benzene, which is used to decaffeinate coffee, for example, can damage DNA by breaking it or cross-linking disparate sections. Our cells are constantly dividing. Many tissue types are routinely exposed to external elements, so it might not surprise you to learn that DNA mutations occur all the time. But you may not realize that according to some estimates, up to 1 million alterations of our DNA occur every day throughout our body. So the question is, how do we stay healthy at all? Thankfully, we have a variety of DNA repair proteins in our cells, constantly monitoring and fixing our DNA. That is, each cell is equipped with strategies for proofreading replicated DNA and repairing any errors. If the DNA errors are too severe, the cell can stop dividing entirely, a process called senescence, or it can commit suicide, known as apoptosis. Unfortunately, in some cases, the mutations are undetected or uncorrected, and the genetic alterations have the opposite effect, supercharging aspects of the cell's physiology and promoting uncontrolled proliferation. In essence, this is how cancer develops. For example, some mutations might cause uncontrolled gene expression, so the gene product is constantly produced instead of shutting off when it's no longer needed. If the resulting protein contributes to the cell's internal control on cell division, for example, it might promote unregulated replication. Other mutations may lead to an overabundance of receptor molecules on the cell's surface. The extra copies of these receptors endow the cell with extra sensitivity to circulating growth factors or hormones, further promoting their proliferation. Both of these mutations, or these errors, can cause the cells to replicate unchecked. But mutations can also silence tumor suppressor genes or promote the expression of cancer-inducing genes called oncogenes. Still, other mutations can alter the expression of cell proteins that the body normally uses to detect aberrant cells, in effect shielding the cancerous cells from detection by our immune system. Today, it is firmly established that cancer is a genetic disease, a disease caused by the occurrence and accumulation of an array of genetic alterations that transform normal, healthy cells into malignant ones. Note that calling it a genetic disease doesn't mean that the environment has no influence. As we've just seen, the environment of the cell has tremendous power. Cancer is still complex and deadly, but advances in genetics are leading a revolution in the understanding and treatment of cancer, so there is much to be hopeful for. Today, cancer cells can be assessed in terms of the genetic mutations that they harbor, and this insight both informs the prognosis of the disease, whether the patient has an aggressive form or one that is more easily treatable, as well as the choice of a precise treatment tailored to that particular tumor. This is a major shift from how cancer has been diagnosed and treated historically. In the past, a diagnosis was made largely by the tissue of origin and by a staging process determined by cellular appearance and presence or absence of metastases, or the evidence of cancer in other parts of the body. So, for instance, a diagnosis of stage 4 breast cancer means that the cancer is no longer limited to the breasts, but has spread and is, at that point, unfortunately, incurable. Oncologists haven't entirely abandoned these techniques, of course, but today the importance of determining the genetic underpinnings of tumors is the cutting edge. So a biopsy, which is a small tissue sample of a possible tumor, is routinely sequenced or stained to determine whether it contains any of the growing list of cancer-causing genetic mutations or alterations in the protein expression levels, which are a consequence of genetic mutations. Knowing these underlying genetic alterations is becoming just as important as knowing the site of origin. Is it in the breast or in the lungs? Or the stage of cancer? Is it stage 3 or 4? For a couple of reasons. First, scientists are realizing that the course and treatability of the disease are influenced by the nature of the mutations and the resulting cellular defects. Some mutations confer resistance to chemotherapy or aggressive disease progression, while others are more responsive. Second, an increasing understanding of the genetics of cancer has facilitated the rise of a more precise medical approach in which drugs that specifically target a known defect can be prescribed to the patients. Historically, without knowing the genetic and molecular basis of a tumor, cancer treatment was centered on chemotherapy. It's highly effective, but it's highly toxic as well, capable of indiscriminately killing any dividing cells, be they cancer cells or healthy ones. Now, knowing a tumor is, say, overexpressing a specific protein, an oncologist can prescribe a drug that blocks just that protein, thereby disrupting a signaling pathway that the tumor is using to proliferate. You'll hear more and more about cancer patients being referred to as HER2 positive, for example, which indicates specific genetic mutations identified in the tumor. This is now just as important as knowing that the tumor was found in the pancreas or the liver, since the doctors can modify their treatment decisions accordingly. In most cases, the genetic alterations in tumor biopsies are assessed not at the genetic level, but at the protein level. But the ability to directly sequence our genes is becoming routine. Major academic medical centers in the US and around the world have their own DNA sequencing machines, and they are beginning to use sequencing results to diagnose and inform the clinical care of their patients. About 45,000 people now have had their entire genetic sequence determined, and we expect this number to grow very quickly in the coming years. In the next 20 to 30 years, I think you'll see each of us having our entire genome sequenced several times in our lives as a checkup to monitor for new and potentially serious genetic mutations that arise, say in skin discolorations or are detected in swabs from our mouth or in DNA collected from a blood sample. Yet another way in which heritability is more subject to change than you might think is through gene insertion. Would you believe that viruses have been inserting genes into the human genome throughout our evolutionary past, to the extent that almost 10% of our DNA is of viral origin? It sounds like science fiction, but it's true. These bits of viral DNA are called endogenous viral elements, and they are the consequence of infection by a class of viruses called retroviruses. Retroviruses are viruses that replicate by turning RNA into DNA, a process that is opposite to the DNA to RNA transcription that our cells normally perform. That's why they're known as retro or backward viruses. They perform this molecular rewind with an enzyme called DNA reverse transcriptase. You might be aware that human immunodeficiency virus, or HIV for short, is a type of retrovirus. The retrovirus inserts its own DNA into the host cell's genome using reverse transcription. This way, it piggybacks on the host cell's ability to replicate genes, since the virus can't replicate itself. Obviously, inserting new DNA into the existing sequence will alter the original sequence. If the viral DNA is inserted into the DNA of germline cells, those cells that generate the eggs and sperm needed for reproduction, then those inserted mutations will be transferred to the next generation, locking them into one's genetic lineage. Most of the viral DNA in our bodies is non-coding at this point, meaning that it is no longer used to produce viral proteins. It's just excess genetic baggage. It used to code for more proteins when our ancestors were first infected, but thankfully, over time, random mutations in our ability to repair DNA, remember the 1 million replication mistakes every day, have rendered most of it unusable. Make no mistake, though. As evidenced by the amount of viral DNA found in the DNA of humans and other animals, viruses have been pestering us for millions of years. And I doubt that they will stop anytime soon. Viruses aren't the only source of gene insertion, however. Human beings have also gotten in on the act. Perhaps the most amazing and controversial aspect of modern genetics is the growing toolbox of techniques used to specifically create or isolate and then insert genes into other living organisms. This is taking genetic modification to an entirely new level, a level that allows our species to trump evolution and natural selection and influence our genes directly. In the first lecture on genetics, I noted that there are numerous monogenetic diseases in which the loss or dysfunction of a single gene causes disease, such as sickle cell anemia and cystic fibrosis. Wouldn't it be great if we could fix nature's mistake and repair or replace the defective gene? Such gene therapy also holds potential promise, and some would say peril, of genetically modifying crops and livestock known as GMOs. With a rising global population and finite environmental and financial resources, biotechnology and genetics offer opportunities to create drought and pest-resistant crops, more environmentally friendly and healthy meats and fish, and to provide aid to individuals suffering from a myriad of diseases. Of course, modifying genes is such a major change to our bodies and our ecosystem that it is not without controversy. If we take a historical view, genetic engineering has its roots in the cellular production of replacement proteins like insulin, which originally had to be extracted from the pancreas of pigs that were raised for this purpose. Introducing the human insulin gene into yeast or other single-cell systems has allowed us to create miniature factories in a dish for the production of these valuable and, in some cases, life-saving proteins. But the idea of putting pig cells into humans was, at first, very hard for many people to swallow. Would other porcine features accompany the cells? By now, we know that for insulin, that's absolutely not the case. But many farmers who would benefit from GMO crops still worry that, for example, inserting a fish gene would make tomatoes taste fishy. In essence, gene therapy requires two critical ingredients: the DNA of the gene of interest and a means of inserting this DNA into the host genome so that it is successfully incorporated and reliably expressed in the cells of the host. In sickle cell disease, it would be inserted in the bone marrow, the source of the red blood cells. In cystic fibrosis, it would be inserted into the lungs. In biological terms, we need a transgene and a vector. A transgene is the gene that will be traveling to the other organism, and the vector is an intermediary used to pass the genetic material into the host organism. In animal studies, transgenes can be introduced directly into the fertilized eggs of a host species. Some of the recipient egg cells will incorporate the new DNA, resulting in offspring that harbor and express the foreign gene product. This approach is impractical and arguably unethical in humans, so a different strategy is needed. We can't just insert new DNA into an embryo without knowing how it will affect the development of a child. But our sometime friend, the virus, is very efficient at transferring genetic material into host cells. Much to our chagrin, of course, the viruses responsible for the common cold and the flu are highly efficient at invading our bodies, entering specific cell types and hijacking our transcription machinery to replicate their viral DNA or RNA, churning out additional virus copies. Unlike retroviruses, adenoviruses such as cold and flu viruses do not integrate into our chromosomes, so their effect is short-term, not potentially lifelong. As such, they make valuable vectors for inserting desirable DNA. Ideally, gene therapy proceeds in 3 stages. A single treatment delivers the repaired gene, and then the gene is incorporated widely into the tissue of interest. And finally, the transgene is expressed for long periods, perhaps even a lifetime. In 2012, the first gene therapy in the Western world was approved, an adenovirus-based treatment for a rare but life-threatening disorder called lipoprotein lipase deficiency. Individuals affected with this disease are unable to produce an enzyme involved in fat metabolism due to a deficit in a gene. As a result, they eventually suffer a variety of severe symptoms, like potentially lethal inflammation of the pancreas. The new treatment uses an adenovirus engineered to deliver the DNA for the gene directly into the patient's muscle and generate sustained levels of the needed enzyme. The therapeutic effects have been significant and long-lasting. I have no doubt that similar gene therapies will be tested for other genetic defects in the coming years. The accelerated speed of our genetic discoveries and advances has outpaced our ethical understanding of the consequences of applying this technology. Promising as our progress is, for some people, it also calls to mind the nightmare vision of Aldous Huxley's Brave New World. In February of 1975, a group of pioneering scientists gathered near Pebble Beach, California, to discuss the ethics of genetic engineering and, importantly, lay some ground rules for the reasonable and prudent advancement of this new and powerful science. But with all the technologies now available to us, we need to revisit and refine these guidelines. We need to emphasize the importance of bioethics, particularly when it comes to genetic engineering in humans. But if we can insert foreign DNA into humans for therapeutic purposes, then surely we can insert foreign DNA into other organisms as well. And indeed, this is very possible. You might have heard of the Flavor Saver tomato, a tomato that didn't spoil as quickly as native varieties, or of Roundup-ready crops, which are equipped with a herbicide-resistant gene from bacteria, allowing them to thrive in the presence of the herbicide called Roundup. Most of the soybeans grown today have this bacterial gene. Societal responses to genetic engineering have been mixed. With regards to GMOs, fears of adverse effects on human health persist, from apples to rice to salmon. It's important to distinguish valid concerns from uninformed fears, however. If we insert a bacterial gene into a soybean, isn't that like eating the bacteria? It's not. Mother Nature is thrifty, as you know, and similar but distinct genes called homologs, as we talked about earlier in the evolution lectures, are found across unrelated species. One gene does not a genome make, and inserting or replacing a useful gene from one species into another is not akin to transferring the essence of the donor species. It's not a Frankenstein's monster. But there are nefarious issues related to GMO technology, with some large corporations claiming trademarks on seeds and charging people who grow them by accident when the seeds spread in the wind, for example. Just by the force of nature, plant seeds can cross fences and barriers. If your neighbor is growing Roundup-ready plants and a few of his seeds pollinate your plants, the corporation that engineered the seeds doesn't have a legitimate claim against you. You didn't ask for the seeds, so why should you pay for them? And yet, the corporations often go after farmers in exactly this type of predicament. My hope is that society will adopt a rational approach to improving and adopting genetic engineering, one that balances acceptable risk and reward. Otherwise, we risk succumbing to the devil we don't know instead of addressing the devils we do. Understanding the implications of our discoveries in terms of their effect on society has never been more critical.